Hi, so today my poster is going to be on a database called DbCAN-PUL that we developed. So this is a database of experimentally characterized CAZyme gene clusters and their substrates. So PUL, or polysaccharide utilization loci, are discrete gene clusters of CAZymes and their genes that work together to digest and utilize carbohydrate substrates. We've developed an online database, DbCAN-PUL, to display experimentally verified CAZyme containing PULs from literature with associated metadata, sequences, and annotation. So we manually collected and curated data from literature by performing 2 rounds of searches against the PubMed database using the first round a general query and a second round a query that includes specific substrates. So the general query will include keywords like saccharide, polysaccharide, carbohydrate, utilization, catabolism, operon, cluster, locus, etc. The second query would be the list of substrate keywords listed here in this table. So these specific queries do not mean to be the most complete. Instead, it's primarily used to complement the general query. So papers from that were hit from the queries were retrieved and manually curated based on the following criteria, 1, 2, 3 listed here. So a total of 1,113 papers were yielded from both rounds of PubMed searches. And after manual curation, only 294 papers remain meeting our selection criteria. So the content comparison of PULDB, a well-recognized published PUL database, and our DbCAN-PUL can be reflected in this table here. Additionally, there's an overlap between PULs of our DbCAN-PUL and those of PULDB. Approximately 469 PULs from PULDB were literature curated, but they're not necessarily experimentally verified or with a substrate characterized. So in order to standardize the signature gene annotation in PULs, including CAZymes, transporter, transcription factors, and signal transduction proteins, all the PULs in DbCAN-PUL were subsequently ran with DbCAN's CGC Finder, a tool to screen for CGCs. So CGCs, or CAZyme gene clusters, are a term that we developed back in 2017, I believe. So CGCs are a border term that is intended for the study of CAZyme gene clusters in genomes without implications on their functions or target substrate. So the predicted CGCs from CGC Finder are therefore not necessarily identical to the PULs from our DbCAN-PUL, which were collected from literature, in terms of genomic boundaries. So this created 4 scenarios to the predicted CGCs on the PULs shown here. And out of the 602 PULs in our DbCAN-PUL database, 524 have CGC predicted, and 78 PULs with the CAZymes, but without CGCs predicted. They might be due to CGC Finder not being able to find its required signature genes, or it may be due to the limitations of our CGC Finder. So we've also developed a user-friendly website for PUL, for DbCAN-PUL. For each PUL, the PUL annotation page has 5 information tabs for the PUL. They include general information, literature information, genomic location information, CGC Finder results, and homologous loci. So you can navigate to PULs according to different categories. So in C1 here, you can navigate according to substrates and taxonomic genera, and even methods of characterization. And if you want to take the extra step and browse the database through a more detailed taxonomic approach, we also made a taxonomic page by Krona. So just click on the taxonomic group you wish to browse, and the server will simply take you there. So in conclusion, DbCAN-PUL offers a comprehensive repository of functionally characterized PULs with rich metadata and we believe it will be a valuable tool and resource for current and future CAZyme research. Thank you.